So here in 11.3, we are going to be taking a look at RNA transcription. So we've taken a look at replication, and we looked at replication in bacteria, and we looked at replication in eukaryotes. We're going to do the same thing for transcription, although they're quite similar. We're going to take a look at the differences as well. So let's take a look at transcription in bacteria. So, as a reminder, information in DNA is used to make RNA. Right? And so that's what we're doing is we're transcribing, we're scribing, writing it out. We're using DNA to make a copy, which is our RNA. And remember that in DNA we have T, which is thymine, and that is replaced with U, which is uracil in RNA. So in bacteria, our first enzyme is RNA polymerase. So our RNA polymerase is what we find in bacteria. We don't have RNA, we have different RNA polymerases in eukaryotes. We have 1, 2, and 3, but in bacteria we have just generalized RNA polymerase. And this is going to use the antisense strand of the DNA as a template. Our RNA polymerase is going to consist of 6 core polypeptide units, including one that's called the sigma unit. And this is going to be important when we talk about transcription, but then this is also going to become important when we talk about things like antibiotics and disinfectants and things like that. So it's important that you understand that our RNA polymerase consists of a core of 6 polypeptide units, and then one is the sigma unit. So our RNA polymerase is going to add complementary RNA nucleotides to the 3' end of the growing strand. So adds complementary RNA nucleotides to 3' end of the growing strand. So recall that we have our DNA, which is 5' to 3', and then we have our other strand of DNA, which is 3' to 5', and we have our various nucleotides that are sticking up in here. Then what we have is our RNA polymerase that's going to come along and attach to the entire complex here. And this, again, is going to consist of those 6 polypeptide units, including one sigma unit. So it's going to attach to the DNA, and then it's going to move down the DNA. It's going to use the antisense strand, so the antisense strand here, to make the RNA. So it's going to be utilizing the nucleotides here. Let's say A, T, C, G, and on and on and on and on. And then it's going to be adding our opposites here. So our U, our A, to a growing strand here. G, C, and on that way. So we have our giant, this red thing here is supposed to be the RNA polymerase. Now, it's not just a matter of it going in there and starting. So let's take a look at what's called initiation. So initiation includes what's called a promoter region. And the promoter region is a specific DNA sequence. This is where the RNA polymerase is going to bind during the initiation of transcription. And so this is where we see that sigma subunit of that RNA polymerase. That's where it's going to bind. So we're going to take a closer look at the promoter region and what's called the initiation site. So we're going to draw in our promoter region here. So I'm going to kind of just put in kind of this box here. This is going to be our promoter region. So we've kind of already gone past the promoter region if we think about it here. But anyway, so our promoter region is going to be what's called upstream. So let's write some of these terms down here. So we have upstream and we have downstream. So upstream is going to be preceding the initiation site. So this is where the promoter is located. For the gene it regulates, right? Because the promoter is what's going to initiate transcription. So that means it's going to be part of regulating whether or not that gene is created or not, right? Whether or not our RNA is made based on that gene sequence. And we'll talk a lot more about that in future chapters, but the promoter region is going to be upstream, so preceding where we actually start. So the initiation site is where the first nucleotide is matched. So the first nucleotide is matched. So we have the promoter region that is going to allow for the attachment of our RNA polymerase. Then we're going to lay down our first nucleotide at the initiation site. So upstream to the initiation site, preceding it, is where we have our promoter. Then downstream is where we have our nucleotides that are following the initiation site. So those are two terms. So our upstream and downstream. So upstream, and both of these are related to the initiation site. So we have our initiation site where we put down our first nucleotide. Upstream is going to be previous to that, and downstream is going to be after that. So previous to that, or upstream, is where we have our promoter region. And our promoter region is going to consist of two different parts here. So we have at the -10 position, -10 meaning upstream, or 10 previous to the initiation site. So 10 nucleotides before where we lay down our first nucleotide is where we have our TATA box. So it's part of our promoter sequence, and it's the TATA box, as it's called, which is TATAAT. So it's this sequence here. We call it the TATA box. The TATAAT is the actual order. And so this is at the -10 position. So 10 nucleotides upstream from the initiation site where we put down our first nucleotide for that particular RNA sequence, we have the TATA box. And the TATA box is important because this is where we are going to have our RNA polymerase attached. So part of these subunits, the RNA polymerase subunits, are going to attach here for initiation. Now, that one last subunit that we talked about, that sigma subunit, is going to actually attach at the -35 position. So this is also a promoter sequence. And this one is going to bind that sigma subunit. So what we see is that at the -10 position, we have all of these proteins, or core proteins of that RNA polymerase, that are going to attach, these subunits attached to the -10 position. Then what we see is this additional subunit, the sigma subunit, is specifically going to reach out, or however you want to say that, but reach out to the -35 position and attach there. So once those two things have happened, then our RNA polymerase is in the correct position to start going through transcription, so to go through the elongation period. So again, we're in the initiation period here, which includes the initiation site, which I did not spell correctly here. So initiation site, try to talk and write at the same time. The initiation site as well as the promoter region. So if we went back up to our image here, this is our promoter region. So we would say at -10, we would write in our TATA box here. And then upstream at our -35, this is kind of ... because I'm not going to write 35 up. Then we're going to have more of our promoter region. And this isn't a specific region. This is going to be different depending on the different types of sigma subunits, which we'll talk about at a later time. But the TATA box is something that we see in all of these organisms here, and all of the bacteria. We have the TATA box. We even have the TATA box in eukaryotic organisms as well. So we have the TATA box. We're going to attach some subunits here. We attach the sigma subunit here. Then our RNA polymerase is all set to go. Then it's going to start making its way down. It's going to lay down its first nucleotide, opposite the DNA at the initiation site. So that's our first nucleotide. And that would be then the beginning of our transcript, which I have here as kind of moving forward. So that is our initiation. Then we have elongation. So elongation is then going to start when that sigma subunit that's at the -35 position is going to dissociate from the RNA polymerase. So it's going to break off from RNA polymerase. So sigma subunit dissociates from RNA polymerase. So it's done its job, it's attached there, it's stabilized RNA polymerase. Now that's going to break off, and then as elongation sounds like it is, then our nucleotides are added to the 3' end to build the strand. So elongation is just that addition of all of those nucleotides, all of those RNA nucleotides. Remember the U instead of T, for example. But we no longer have that sigma subunit. We just have the rest of the subunits as part of the RNA polymerase that's going to continue moving down the DNA, making that RNA transcript, until we have termination. In termination, we actually have what are called termination signals. And termination signals are really just repeated nucleotide sequences. So in the DNA itself, we have the same nucleotides over and over and over again. And what this does is it causes that RNA polymerase to kind of stall. It confuses it because it has so many of the same nucleotides. It kind of like loses its place and isn't sure what's happening. So then it ends up falling off. So it stalls and then it falls off. And that's how we actually end transcription in bacteria. All right, so that is our termination, our repeated sequences at the end, and then it causes it to stall and causes it to fall off. So now we're going to take a moment to compare that to transcription in eukaryotes. So our transcription in eukaryotes. And we're just going to compare the two. So for transcription in eukaryotes, we have 3 different polymerases. We have polymerase 1, we have 2, and we have polymerase 3. So these are DNA, or not DNA rather, but RNA polymerases. So RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Whereas, as we mentioned in bacteria, we'll say prokaryotes, we have just RNA polymerase. And then, of course, that includes those 6 subunits, however. Our eukaryotic organisms are also what are called monocistronic. So monocistronic. And what that means is that the mRNAs that are made, those mRNA transcripts that are being made in transcription from the DNA, code for only one protein. So when we have studied transcription previously, for example, perhaps you're used to thinking that we're transcribing one gene, and that one gene is turning into one mRNA, via transcription, and then that one mRNA is going to, leave the nucleus and go make a protein. And that's true for eukaryotic organisms. But in bacteria and archaea, so in prokaryotic organisms, they are polycistronic. And polycistronic meaning that the mRNAs code for more than one protein, and can be many. And not only do they code for more than one protein, but all of the proteins that are encoded in that mRNA have related functions. So this is another way that prokaryotic organisms are very, very efficient. So if we have our mRNA transcript or RNA transcript in a prokaryotic organism, we would have a gene, let's say a gene here, a gene here, a gene here, a gene here, and then each of these sections here on the mRNA or on the RNA are going to code for different proteins, but those proteins likely will then come together and make the finished product. Versus in eukaryotic organisms, if we have this same long mRNA, then we would just have this entire thing here is coding for the one protein. So it only codes for one protein. We have one mRNA for one protein, versus in prokaryotic organisms, they make one long protein, or one long mRNA rather, that have several proteins that all have related functions. So they're all made at the same time, all together. They don't have to make multiple mRNAs to have multiple proteins. The last difference that we'll mention is that in eukaryotes, we have RNA processing. And so remember that RNA processing includes, we have that pre-mRNA. So pre-mRNA. And then what happens is within the nucleus, and this is because it's nucleus bound. Nucleus bound. We have to add that 5' cap. We have to have that 3' poly-A tail. And then we also have RNA splicing. And then we have our finished mRNA. So we have a pre-mRNA, it then gets a 5' cap added to it, a 3' poly-A tail, goes through RNA splicing, where we cut it into pieces. We have the exons and the introns, and we're going to glue those exons back together. And then we have our finished mRNA. That finished mRNA is then going to leave the nucleus through a nuclear pore and go out into the cytoplasm. So we do not have RNA processing. So in prokaryotes, there is no RNA processing. And that's because we're already in the cytoplasm, right? Because we don't have a nucleus in our prokaryotic organism. So it's already out there. We add a 5' cap and a 3' poly-A tail, for example, in eukaryotes, because as we send that mRNA out into the cytoplasm, we've got lots of different enzymes that are out there that want to eat it up and break it down. And we add the 5' cap and the poly-A tail. The 5' cap is used really for transcription purposes, or translation purposes for the binding to the ribosome, but it also protects at getting eaten away by other things. More so, though, the 3' poly-A tail is important for protecting that material in there, protecting that transcript for that gene, because as it's hanging out in the cytoplasm, again, we have these enzymes that are going to start eating up DNA and RNA, and so it's going to start to go in and it's going to start to be clipped off nucleotide by nucleotide. So if we have this long poly-A tail, meaning many A's at the end of our mRNA transcript, then as it's kind of eating that, it's not going to be bothering the actual important information in the middle region there. And then, of course, RNA splicing is so that we can take our pre-mRNA, which we can pretend is up here, we can kind of cut it into pieces, and depending on if we use piece 1, 2, 3, 4, 5, If we use 1 and 3, we can get one protein. If we use 1, 2, and 5, we get a different protein. And so we can chop up the same pre-mRNA and get different proteins. And we don't need that, of course, in our prokaryotic organism because, like we said, it's polycistronic. So it just has all the proteins it needs all in one mRNA. So we don't need this RNA processing. It's already hanging out there. It's already getting eaten up by the same enzymes. But it's okay because it's making many copies of these and it has all of the different proteins on the one mRNA. So those are 3 differences between eukaryotic and prokaryotic transcription. And that wraps up our transcription. So a lot of that should be familiar to you. You can go back and kind of take a look at eukaryotic transcription again if you're not familiar with it. But this is really kind of the bulk of the information you should know for this chapter, or this section.